Welcome everyone, and thank you for joining today's Science AAAS webinar entitled Deciphering Immuno-Oncology, Targeting Cellular Mechanisms of the Tumor Immune Response. I'm Sean Sanders, Director and Senior Editor for Custom Publishing at Science, and I'll be moderating this webinar. Tumors are complex hubs of cellular activity that incorporate immune cells, vasculature, and even bacteria. It has become increasingly clear that a better understanding of this tumor microenvironment and the different types of immune cells within it is critical to developing more effective cancer therapies. In particular, immunotherapies that target the signaling mechanisms of T cells have shown great promise for many patients. However, these techniques have proven successful for only a fraction of people and against just a few cancer types. A detailed dissection of the molecular signaling switches that control immune cell activation and tumor checkpoint evasion is required if we hope to broaden the reach of immunotherapy treatments. Our speakers today will describe key signaling mechanisms and shed light on how they can be utilized to better activate and control immune cells in the tumor microenvironment. It's my pleasure to welcome our speakers today, Dr. Elizabeth Jaffe from Johns Hopkins University in Baltimore, Maryland, and Dr. Matthew Galsky from the Icahn School of Medicine at Mount Sinai in New York City. Thank you so much for joining us today. Before we get started, I wanted to give some important information to our online viewers. You will find photographs of today's speakers in the Presenter tab to the top right of your screen. Just click on the View Bio link to read more details about their background and research. To the right, you will also see the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the presentations, we will have a short Q&A session during which we will address some of the questions submitted by our live online viewers. So if you are joining us live, start thinking about some questions now, and please submit them at any time by clicking the Ask a Question tab. Also to the right, typing the question into the message box and then clicking Submit. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the bottom left under the slide viewer. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Cell Signaling Technology for sponsoring today's webinar. Now I would like to introduce our first speaker, Dr. Elizabeth Jaffe. Dr. Jaffe is Deputy Director of the Sidney Kimmel Comprehensive Cancer Center, Co-Director of the Skip Virag Center for Pancreatic Cancer, and Associate Director of the Bloomberg Kimmel Institute for Cancer Immunotherapy, all at Johns Hopkins University. Her research focus is on developing novel immunotherapeutics for the treatment and prevention of pancreatic cancer. Thank you so much for joining today's webinar, Dr. Jaffe.
Thank you, Sean. It's a pleasure to speak to all of you today. The topic of my talk will be Converging Technologies Uncovering Tumor Microenvironment Crosstalk. I'd like to just share with you my disclosures. Sean, if you could advance the next slide. These are my disclosure information. I am happy to discuss with any of you after the talk if you have any concerns. Next slide, please. I'd like to start with a perspective slide. This is a historic time in cancer research and cancer care. This is accredited to the past 30 years of scientific discoveries that led to new treatments. The recent rapid pace of scientific and clinical research discoveries parallels the technologic developments. And if you take a look at the slides, you could see that we really began starting to discover these immune checkpoint signals or signaling pathways back in 1987. At that time, the technologies we had available to us was PCR. It took about 15 years to move from that point to a point where we were able to also understand the PD-1, PD-L1 pathways, and we were also able to start to develop antibodies that inhibit these pathways and show in mouse models that inhibiting these pathways leads to T-cell activation. This is also about the same time that we sequenced the human genome for the first time, 2000. It took another 10 or 11 years before we saw the first FDA approval in the clinics of these developments. And interestingly, that's about the same time We saw the development of next-generation whole exome sequencing, RNA sequencing, multiplex proteomics, metabolomics, single-cell technologies. And this is important to point out because over the next 8 to 10 years, which brings us to now, we've seen more than 30 approvals of immune checkpoint agents for a number of different indications. In addition, we've also seen the development and approval of CAR T cells. Next slide, please. So this slide just shows you that these immune checkpoint inhibitors are not only working in a number of different cancers, but in some patients, they're very, very durable. And in fact, we now have 5 years follow-up for metastatic melanoma, looking at both anti-CTLA-4, the first approval, and you could see on the right side, the top graph, that's the overall survival. Below that is the progressive-free survival. And in both cases, anti-CTLA-4 does have a pretty good long-term 5-year survival benefit for these patients. Anti-PD-1, the curve right above it, shows that there's an even better response rate. And when you add the two, you have an even longer, almost around 50% 5-year survival. So this is very important because these agents not only are showing regressions of patients' tumors in the metastatic setting, particularly in cases where patients failed other therapies, but importantly, they're very durable and patients have good quality of life. Next slide, please. So this brings me to the point that we're now in a new era of precision immunotherapy. It's now possible with these new technologies to take a small piece of tissue, biopsy a small piece of tissue through using CAT scan visualization or some other visualization radiographically. And with that small piece of tissue, we're able to do many different analyses that allow us to better understand the patient's tumor. So for instance, it's now possible to determine the mutations that a patient's tumor expresses, and by determining the mutations, we can now develop neoantigen vaccines that are patient-specific targeted. And these are already in the clinics, and I think you're going to be seeing a lot more of these type of therapies in the near future in multiple different cancer types. Also, we're able to sequence the T-cell receptor in a patient's T-cell that sees specific antigens, and these can be now genetically engineered to be expressed by lymphocytes from that patient. So we can now have an adoptive therapy that's personalized towards specific antigens. And then, of course, we have CAR T cells where we can not only alter the CAR T cell to express inhibitors or activators of signals on T cells, but we can also deliver antigens that can specifically target the CAR T cell to a particular tumor. Next slide, please. So immune checkpoints have shown unprecedented responses in a number of advanced cancers. And again, this is attributable to a number of different factors. We now know that many of these cancers have a high tumor mutational burden. They also express high levels of PD-L1. And of course, there's availability of tumor-specific T cells that are either in the tumor or in the tumor-draining lymph nodes, and they are activated when you give these anti-targeted agents, anti-signal targeted agents. The problem is not all tumors with these characteristics respond to current immunotherapies, and some cancers that do have these characteristics don't respond to these immunotherapies. Next slide. So two people that I've worked with, Mark Yartwin and Alex Hopkins, wanted to better quantitate the number of mutations or a threshold number of mutations beyond which we could predict which patients with which cancers would respond to immunotherapy. So this was done by looking at all of the published phase 2 and phase 3 studies for anti-PD-1 and anti-PD-L1 therapy. And on the x-axis, you could see the median number of coding somatic mutations per megabase. On the y-axis is the objective response rates. And what you see, if you take a look, is that a minimum of 10 somatic mutations per megabase is required to definitely have an anti-tumor immune response with anti-PD-1 or anti-PD-L1. But what's interesting is that there were a couple of outlier cancers that respond but have lower mutational burdens. Next slide. One of which is Merkel cell. And so Merkel cell is interesting because there are two forms of Merkel cell. There's the viral-related form, and that's on the left side in the red box. And that form actually is a low tumor mutation burden, but because it's virally associated, it appears to attract T cells. On the right side are the UV-induced viral negative tumors, and those do have a higher mutational rate, which we expect because UV induction can induce this high rate, and that's why melanomas are so responsive to immunotherapy. Interestingly, renal cell actually has a different reason why these cancers have about a 25% response rate. Next slide. So it turns out that there's more than one type of neoantigen, as most of you probably realize now. The ones that we've typically looked at in the past have been single nucleotide variants. However, Charlie Swanton's group observed that insertion and deletion, or indels, derived tumor-specific neoantigens may be even more immunogenic, and in fact, in the case of renal cell carcinoma, this appears to be the case. Next slide. So Mark Yarchwin in our group subclassified the tumor microenvironment based on tumor mutation burden and PD-L1 status and reported that there are at least 4 subtypes. The first, all the way on the right, would be your hypermutated and inflamed cancers such as melanoma and cigarette-induced lung cancer. Or smoking-induced lung cancer. And those are the ones that have a high tumor mutation burden and high PD-L1. And when you look at these tumors under the microscope, they have T cells infiltrating. So anti-PD-1 or anti-PD-L1 just activates them, and these tumors typically respond. Then you have the hypermutated. These are ones that have a high tumor mutation burden, but you don't see PD-L1 expressed on a pretreatment biopsy. The reason for this is that the T cells are sitting elsewhere. So the T cells are required to secrete interferon and other cytokines to upregulate these checkpoints. As soon as you give anti-PD-1 or anti-PD-L1, they infiltrate the tumor, upregulate PD-L1, and then you get a tumor-killing effect. The third one is the renal cell situation, where you have inflamed form, and that's tumor mutation burden that's less than 10, but you have high PD-L1, T cells are coming in, and this would be because the neoantigens are not your typical neoantigens, but they're indels or some other form, maybe a fusion protein neoantigen, and so the T cells are in there, and as soon as you give anti-PD-1 or anti-PD-L1, they respond. And then we have the fourth form, which is the not hypermutated and not inflamed, and this is low tumor mutation burden and low or no PD-L1. You can't find a T cell. You give anti-PD-1, anti-PD-L1, anti-CTLA-4, and you still don't see a T cell coming in. And we consider these the immune-resistant form. Next slide. Next slide, please. Still not moving on my screen. I think we bypassed it. There we go. Thank you. So there are a number of challenges to progress in immune-resistant tumors, and that's what I'm going to spend some time talking about for the rest of my time slot. The first is the tumor microenvironment of immunotherapy-resistant cancers have multiple immune suppressive signals, and it may be that we need to bypass multiple signals to see a good immune response. Also, heterogeneity is becoming an important aspect of research that may play a role in these tumors, and within the same patient, there's even going to be heterogeneity among the tumor cells, among the stromal cells, and among the inflammatory cells that are infiltrating. And then we're also learning that it's not just getting a T cell into a tumor is not enough, but it's really the quality of the T cell that needs to be considered when you're trying to induce an immune response. Next slide. So I work in pancreatic cancer, and I think it's a good example of a immune-resistant tumor. And pancreatic tumors, what's interesting about them is that they only have about 10 to 40% tumor cells, and the rest is a stromal reaction. And you're going to hear a lot about the stromal reaction from myself and from Matt, who will follow my talk. And within that stroma are multiple different cell types of fibroblasts that get co-opted by the tumor cells to help support tumor growth. But also inflammatory cells fall into that category as well, and they infiltrate the stroma. And initially they probably come in to clean up the initial problems, the initial mutations that occur. But then they become a chronic inflammation, and they get also co-opted to help the tumor grow. And these are in the form of monocyte macrophages, immature myeloid cells, regulatory T cells, there are regulatory B cells, and then, of course, some of the stromal cells lay down extracellular matrix that can be inhibitory. But the good news is emerging technologies and analysis platforms are providing us a new opportunity in which we can now begin to understand the complex signaling networks that occur between all of these populations of cells infiltrating these tumors. Next slide. So pancreatic cancer is unique in that the majority of patients, over 90%, all begin with one mutation, and that's mutated KRAS. And it's mutated KRAS is very interesting because it causes a lot of chaos in the normal tissue, leading to malignancy. Mutated KRAS signals through metabolic reprogramming. It has a role in uncontrolled proliferation. It helps form tumor stroma, which you'll hear more about. And, of course, it also helps form immune suppression. It initially attracts through the expression of GM-CSF and IL-8 monocytes. Those monocytes come in thinking they're going to react to this new mutation, and instead they get co-opted, and with other signals such as IL-6 and TGF-beta and VEGF and arginine, these cells become suppressive. They become either your tumor-associated macrophages or your myeloid-derived suppressor cells. Next slide. So we also know that the tumor genotype and phenotype can now shape the tumor microenvironments. I told you how mutated KRAS does that, but there are other mutations that also lead to shaping this immune-prohibitive environment. So for instance, if you look all the way to the left, the p53 loss in tumor stroma has been shown to turn M1 macrophages, which help in activating a T cell, into M2 macrophages, which suppress T cells. Moving over a little bit towards the right, you see the BRAF mutation. And BRAF mutations do a number of things. They can express IL-1 alpha and beta, which will then help to co-opt fibroblasts to become cancer-associated fibroblasts. They also secrete IL-10, VEGF, IL-6 to help induce tolerogenic dendritic cells. And then all the way to the right, you could see that another observation that many of us have reported is that as you get more suppression, these myeloid cells become the predominant cell type, and the CD8 T cells start to either leave or end up getting tolerized. And so typically you turn a high T cell, low myeloid environment into the opposite, which becomes a high myeloid, low T cell environment. Next slide. So it's also important to point out that these tumors don't start out excluding effector T cells, and in fact it's the accumulation of these genetic and epigenetic alterations that lead to acquired immune privilege, and that's what I'm showing you here. You have your normal cell, and as soon as you get your first mutation, this sets up an inflammatory response. As I mentioned, mutated KRAS initially is more of an alerting signal, a danger signal, and it's going to bring in initially innate cells such as monocytes and T cells. And we are finding that T cells do begin to infiltrate the early pre-malignancy such as PANN1. These T cells have the ability to react against mutated KRAS and even maybe subsequent genetic alterations. But as additional genetic alterations accumulate, this acute inflammation turns into chronic inflammation, and then you have reprogramming of these monocytes into suppressive cells, and you get a reduction in your CD8 T cell. So this is important to point out because there may be earlier time points where you may want to intercept the development of a cancer at a time when the immune system, even in these what we call immune-resistant cancers, are immune-susceptible pre-malignancies. Next slide. So the prior focus has been on tumor and immune cell suppressive mechanisms within tumors, but tumor and immune suppressive cells interact with stromal cells to form a T-cell excluding complex network of signals. And as I mentioned, tumor cells can co-opt normal stromal cells to support cancer progression metastasis. And it's with all these new technologies, we're now elucidating the role of different subpopulations of stromal cells in contributing to immune evasion. Next slide. So what this figure just shows, it's from a really nice review in Nature Reviews Gastroenterology and Hepatology. And it shows the role of stromal cells in the tumor microenvironment and points out the complex interactions between cancer cells, stromal cells, and immune cells. But I'll leave it to Dr. Matt Galsky to delve more into the biology of the different stromal components. Next slide. And what this is just, I think you passed one. That's fine. So what this is showing is just, again, the stromal interactions, right? So, okay, and this is the origin of cancer-associated fibroblasts. And so basically this just shows you that stromal cells or fibroblasts come from different compartments within the body, including the tissue itself, but also from bone marrow, can be attracted from bone marrow. And again, they can transdifferentiate into these cancer-associated fibroblasts. The local fibroblasts can be converted through signaling to cancer-associated fibroblasts. And even these bone marrow stem cell-like cells can be recruited and converted to cancer-associated fibroblasts. Next slide. So what this slide is showing you is that stroma forms the majority of the pancreatic tumor mass. And this tumor mass is home to many different subtypes of fibroblasts, including, as we've been talking about, the cancer-associated fibroblasts, but there's also stellate cells and myofibroblasts. And these are all normal cells, again, that are co-opted functionally to support cancer development, cancer progression, and also cancer metastasis. Next slide. So just a few key points. CAFs include all fibroblasts in the tumor and support cancer progression. They're heterogeneous and include subpopulations arising from distinct cellular origins, such as local fibroblasts and mesenchymal stem cells. There's distinct CAF populations that influence cancer cell proliferation, immunity, angiogenesis, extracellular matrix remodeling, and metastasis. They have multiple functions, both tumor promoting, but if they're reprogrammed, they can also be tumor retarding. So you don't want to deplete these fibroblasts, you want to reprogram them. Next slide. So this just shows you what the function of the CAFs are, and again, you'll hear more about this from Matt. Next slide. So one thing I wanted to point out is that the stroma helps to form heterogeneity within tumors, and this was best illustrated in a recent publication from David Ting. And what this really shows, to put into context, is what happens in the clinic. So in the clinic, I can have two patients come into my office with pancreatic cancer. They both have the same burden of disease in the same sites. They have the same biomarker predicting burden, so the same number measuring that burden. They feel the same. They look the same. Under the microscope, their tumor looks the same. One will respond to chemotherapy and live 1.5 years. The other will not respond to chemotherapy and die within 3 months. So this paper was the first to suggest that the stromal compartment is really helping to determine heterogeneity. And what he did was he did single-cell analysis between cancer cells and stromal cells. And what he found was that within the cancer cells, there are 4 subtypes. You have the ones that are proliferating, Ki67 positive. You have the ones that are undergoing EMT. You have double positives and double negatives. But what was important for heterogeneity is that it depended on the fibroblast components surrounding the types of cells that led to whether the cancer would be aggressive or not. Next slide. So just to summarize, what he found was most aggressive cancer cells had the double phenotype, both proliferation and epithelial to mesenchymal transition markers. And they also had high cancer-associated fibroblasts that were linked with a double phenotype, and this was enriched in MAPK and STAT3 co-signaling. And the CAFs contributed to this heterogeneity. And intratumoral gland types, and he had 8 of them, provided tissue heterogeneity links to clinical outcomes. Next slide. And these are just the 8 different stromal architectural glands. Next slide. And this is the important point as well. So if you looked at the tumors pre-treatment in the patients he looked at, they had a mixture of the different stromal gland forms. But with FOLFIRINOX, the standard of care after treatment, the people who had resistance disease ended up with only 2 forms, and both of those had double positive cells, tumor cells, with either medium or high levels of stroma surrounding those cells. So again, it's the start of thinking about heterogeneity and predicting clinical outcomes. Next slide. So I'm not going to focus on this, but this was another example of being able, using stromal evaluation to determine patient response to cancer immunotherapy, and I believe Matt will be talking more about this. Next slide. And this final slide basically of data shows you that, in fact, there is these LRRC15 positive CAP signature, that if these CAPs have this signature, patients are unlikely to respond to anti-PD-L1 therapy, which I think is very important. Next slide. So the final 2 slides just show you that we now have agents in the clinic that can facilitate both myeloid reprogramming and stromal reprogramming. This shows you the myeloid reprogramming targeting different signals on the suppressive myeloid cell populations. And next slide. And this next slide shows you that there are a number of manipulations we could do to alter cancer-associated fibroblasts and reconvert them to ones that can be anti-cancer. And I'll stop there.
Wonderful. Thank you so much, Dr. Jaffe. We're going to move right along to our second speaker for today, and that is Dr. Matthew Galsky. Dr. Galsky is a medical oncologist at the Mount Sinai School of Medicine and the Tisch Cancer Institute, where he currently serves as Professor of Medicine, Director of Genitourinary Medical Oncology, and co-director of the Center of Excellence for Bladder Cancer. His research efforts involve team science-based approaches focused on dissecting the mechanistic underpinnings of response and resistance to immunotherapeutic strategies in bladder cancer. A very warm welcome to you, Dr. Galsky.
Thank you, Sean. It's really my pleasure to speak to you all today. So as mentioned, I'm a medical oncologist, and my clinical and research focus is on bladder cancer. So a lot of the data that I'm going to describe today relates to bladder cancer, but I do think the lessons are applicable to most tumor types. As with many solid tumors, and we've heard about this already from Dr. Jaffe, immune checkpoint inhibitors have really been transformative for the treatment of metastatic bladder cancer. But also, like in many other solid tumors, these drugs weren't approved by regulatory agencies because the majority of patients benefit from treatment, but rather a small subset of patients responded to these therapies with regression of metastatic disease. But when those responses occur, they tend to be quite prolonged. And this is an example of a patient of mine. These are CAT scan images at baseline. And you can see the arrows pointing to metastatic lymphadenopathy, lymph node metastases from bladder cancer in a patient who had already received chemotherapy and had progression of their cancer. They were treated pretty early on a clinical trial with PD-L1 blockade and rapidly achieved a complete clinical response, clinical complete response, which means to us disappearance of abnormalities on a CAT scan, and has been maintained in that complete response now for over 5 years. So this really leads to the obvious question, why do some patients have these dramatic responses and why do some patients not, and how can we achieve these responses in a higher proportion of patients? So lots of groups have been working hard on this topic for many years, and again, we've heard a lot about this work from Dr. Jaffe already. A culmination of the work really does suggest the pre-existing hypothesis regarding immune checkpoint blockade, which was that pre-existing immunity in tumors enriches for responses to these treatments. And that can be measured in the clinic in a variety of ways, and shown on this slide is data from a cohort of patients with metastatic bladder cancer who were treated with the PD-1 inhibitor nivolumab. And you can see in one panel data regarding T cells based on CD8 immunohistochemistry infiltrating the tumor specimens, and in the other panel, interferon gamma gene signatures. And basically what both of these panels are showing is that the likelihood of achieving a complete or partial regression of cancer is higher with these treatments if there's either pre-existing T cells in the tumors measured by immunohistochemistry or an increase in these interferon gamma gene signatures. So pre-existing immunity enriches for response, and this really begs the question, what other parameters drive response or resistance? So we've been working on this question for a while, as have many groups. And we initially approached the question in a pretty similar manner to some other groups at the time, which was that we had access to gene expression data from a cohort of patients with metastatic bladder cancer. What we did was infer T-cell infiltration using that gene expression data, and then ask a very simple question. Could we identify genes or pathways that were enriched in these specimens that lacked T-cell infiltration in hopes of identifying mechanisms of resistance that might be modulated therapeutically? And so what we found, at least initially, was a little bit surprising to us, because we found some pathways that were inversely correlated with inferred T-cell infiltration. But one of the sets of genes that was enriched in tumors that had a high level of T-cell infiltration was a set of genes that were enriched in the hallmark EMT gene signature. And this was initially a little bit surprising to us, but this was around the time when some of the larger single-cell transcriptomic papers in human cancers were being published, in cancers like head and neck cancer and colon cancer. In those papers were suggesting that the more mesenchymal molecular subtypes of those cancers that had previously been defined by bulk gene expression data probably didn't reflect the biological process of EMT, or at least not solely the biological process of EMT, but probably rather represented the presence of cancer-associated fibroblasts that were accounting for these EMT-related transcripts. And so we used some computational techniques and leveraged a set of patient-derived xenograft models that we had access to, where one can more easily identify the source of epithelial versus stromal transcripts based on whether or not they're human or mouse origin. And we came to a similar conclusion, that these EMT-related genes were really emanating from cancer-associated fibroblasts. And that probably in part explains why we see these traveling with T-cell related gene expression. This isn't that surprising when you dig down a little bit deeper and look at the actual genes, and a lot of the genes in these signatures encode for extracellular matrix related proteins. So what does this all have to do with immune checkpoint blockade? Well, despite the fact that our T-cell and stromal gene signatures were positively correlated, we reason that these signatures might confer disparate predictive or prognostic information if the stromal signatures really represented the presence of cancer-associated fibroblasts. So we looked at these signatures in baseline tumors from a large cohort of patients with metastatic bladder cancer treated with the PD-1 inhibitor nivolumab, and what we found was a significant statistical interaction between the T-cell infiltration gene signatures and the stromal signatures, such that in tumors with low levels of T-cell infiltration, the stromal signature really didn't have much of an impact on likelihood of response or survival. But in T-cell infiltrated tumors, a higher stromal gene signature was associated with a lower likelihood of response to treatment and inferior survival compared to these T-cell infiltrated tumors with low stromal signatures. And for illustrative purposes on the slide here, we just showed the Kaplan-Meier curves with these gene signatures cut at the medians. And so why do CAFs result in resistance to immune checkpoint blockade? And we've heard about this from Dr. Jaffe already. There's a growing recognition, of course, that fibroblasts and the TME are not innocent bystanders, but rather potentially major orchestrators of tumor pathogenesis. And really, our knowledge of cancer-associated fibroblasts is still in its infancy, and a lot of what we know about fibroblast function is derived from the wound healing literature. But probably most of the characteristics of CAFs shown on this slide probably contribute in some way to resistance to anti-tumor immunity. But really, I'm going to focus on the matrixome and the secretome of CAFs because that's really where a lot of the data has been pointing, at least in the past couple of years. So when we saw this statistical interaction between these T-cell signatures and these stromal signatures, in our data set, it reminded us of a spatial relationship between these cellular compartments that's been observed for quite a long time. And that is, and this is almost certainly an oversimplification, but I do think this is a useful conceptual framework. That is that there are these three major tumor immune phenotypes that have been described. There are these tumors that lack T cells and commonly are called immune deserts, and you see these highlighted in this cartoon and also in the bladder cancer specimen micrograph is shown. Then there are these tumors where there's inflammation with T cells, and the T cells are really infiltrating nests of cancer cells. And this is often what's referred to as these inflamed tumors. But there's this intermediate, and that's shown in the middle of the slide here. And that's where there are T cells infiltrating the specimen, but they're really restricted to stromal regions, and they don't infiltrate nests of cancer cells. And so we hypothesized that what we were seeing based on gene expression really was recapitulating these tumor immune phenotypes. And indeed, that was the case when you look at our high T-cell infiltration signature and high stromal signature. The specimens that express both really correlated with these tumors where the T-cells were present based on immunohistochemistry restricted to stromal regions. Importantly, the same year that this data was published, there was this beautiful paper published from scientists at Genentech also highlighting the role of stromal elements in mediating resistance to the PD-L1 inhibitor atezolizumab, also in a large cohort of patients with metastatic bladder cancer. In their paper, they assessed these three tumor immune phenotypes across this large cohort of patients with metastatic bladder cancer, and quite interestingly showed that this excluded phenotype was present in 47% of bladder cancers. So this is probably not a minor mechanism of resistance by any means. And I've seen subsequent work from the group showing this across various tumor types, and not unexpectedly, you see this distribution of these tumor immune phenotypes vary tremendously across tumor types. So this work, of course, demonstrates an association with lack of response to immune checkpoint blockade, but certainly doesn't establish causality or mechanism. And there are some potential clues as to what might be going on from some other groups. And this is a paper from Dr. Helen Salmon and colleagues where they combined immunostaining and real-time imaging of viable slices of human lung tumors. And you can see staining for fibronectin in these viable lung cancer specimens and staining for CD3. And then what they could do is trace the migration of these individual T cells within this microenvironment. And what they showed really beautifully was that the density and orientation of the stromal extracellular matrix likely plays a major role in controlling migration of T cells, suggesting that this dense extracellular matrix may be in part related to lack of response to immune checkpoint blockade just because of physical constraint reasons, because these T cells can't get to where they need to be. The group from Genentech suggested that this process might be driven by TGF-beta emanating from tumor cells or elsewhere in the microenvironment, activating fibroblasts to produce extracellular matrix, with subsequent trapping of T cells in the stromal regions. And in their paper, they show that TGF-beta related gene signatures are associated with lower likelihood of response and inferior survival with the PD-L1 inhibitor atezolizumab compared to tumors with lower levels of TGF-beta signaling. This probably isn't a bladder cancer specific phenomenon, of course. in the D. Carvajal laboratory, in a paper in Nature Communications in 2018, identified an extracellular matrix transcriptional signature that was upregulated across a variety of cancers. And they went on to show that this signature was associated with resistance to immune checkpoint blockade across various tumors, including melanoma. And so here you can see in this heat map this extracellular matrix signature related to TGF-beta signaling, and you can see in the tumors that cluster with an increased expression of this signature. At the bars horizontally across the very top, those black shading of the bars indicate lack of benefit or lack of response to immune checkpoint blockade. And visually, you can certainly see this clustering of this lack of response in these tumors where you have increase in this extracellular matrix-related signature. The matrixome almost certainly plays a role, but the secretome of CAFs is clearly critically important as well. And not only are CAFs activated by TGF-beta and other growth factors, but they're also a rich source for these cytokines and chemokines, and that could result in a potential impact on anti-tumor immunity by directly impairing T cells, but also, of course, these signals recruit myeloid cells, which then can subsequently impair T cell function. There's a very interesting paper from Pieranzoni and colleagues that really links this whole story together, and this paper used a similar approach to the paper that I've mentioned from Dr. Salmon's group, tracing the migration of T cells in viable lung cancer specimens, and showed that it was actually the myeloid cells in the microenvironment that were making contacts with the T cells that were restricting migration in the microenvironment. So this is probably all related to cellular crosstalk within the microenvironment, probably not one mechanism accounting for resistance, but multiple mechanisms accounting for resistance. So these cells are clearly bad, so how about we just get rid of them? And this was tried in model systems, and in this landmark paper where a transgenic murine model of pancreatic cancer was generated in which alpha-smooth muscle actin expressing myofibroblasts could be deleted, you can see what happens when they did that. With deletion of these alpha-SMA expressing cells, you actually have inferior survival of these mice. And so it's not that simple as just depleting a whole population of cells, as alluded to by Dr. Jaffe, and we certainly have to understand the heterogeneity of cancer-associated fibroblasts and the cellular crosstalk in a much more detailed way. Targeting TGF-beta might be a more tractable approach, and the group from Genentech showed that the EMT6 model of breast cancer demonstrated this excluded immune phenotype where T cells are restricted to the stromal region. So they thought this would be a good model to test whether or not this could be overcome with anti-TGF-beta. And you can see in panel B here, in this model, anti-PD-L1 doesn't have much activity. Anti-TGF-beta has no activity as monotherapy. But when you give the two together, you have regressions in a subset of mice. And pharmacodynamically, as shown in panel D, this corresponds to migration of T cells into areas of cancer cells away from the stromal restriction, accomplishing pharmacodynamically what one would hypothesize would happen in this context. And so these trials with immune checkpoint blockade and anti-TGF-beta are moving forward. There's already been some data generated and studies going on across different tumor types. But certainly TGF-beta is pleiotropic. And while this might offer a benefit in some patients, we still really need to understand more about the biology and heterogeneity of cancer-associated fibroblasts to really exploit this biology and overcome resistance. And just in the past few years, there's been an explosion of knowledge regarding subsets of cancer-associated fibroblasts, really driven by the technologies that Dr. Jaffe alluded to, including single-cell sequencing. A lot of the data, though, has been a mix of data generated in mice and data generated in humans, using slightly different technologies, using slightly different approaches, and so there are a number of inconsistencies in the literature, but there are some major themes that have developed. And one of the major themes is that there's probably at least two major subtypes of cancer-associated fibroblasts. These myofibroblastic calves, which are characterized by alpha smooth muscle actin expression, low expression of inflammatory mediators, tend to be higher expression of extracellular matrix. These tend to be located in closer proximity to cancer cells. And then there are these ICAFs, or inflammatory cancer-associated fibroblasts, which express low levels of alpha SMA, high levels of inflammatory mediators. In the secretome of ICAFs differs a little bit from study to study, but the cytokines and chemokines shown on this slide are pretty common across different data sets. So a really nice paper from the Turley Laboratory, described by Dr. Jaffe, used single-cell transcriptomic data from murine models of pancreatic cancer and from human pancreatic cancer specimens, and they revealed that these two separate major fibroblast subsets, which are generally consistent with what's been described for these MyCAFs and ICAFs, probably co-evolved during tumor progression, driven by either TGF-beta or by IL-1. So TGF-beta, closer proximity to cancer cells driving MyCAFs. IL-1, maybe these cells are further away from the epithelial cancer cells, driven by IL-1 generating these ICAFs. And so understanding the heterogeneity and the signaling that leads to this heterogeneity has really led to some interesting observations, including the observation that there are these subset of TGF-beta-driven CAFs that can be identified with a gene signature that, when applied to a bulk gene expression data cohort of patients treated with atezolizumab, the PD-L1 inhibitor, can distinguish patients who have a higher likelihood of response to treatment versus a lower likelihood of response in a longer survival versus a shorter survival. So certainly a number of studies and the culmination of data suggesting that measuring CAF signals in the tumor microenvironment can help us identify, at least prognostically, patients who might do better versus patients who might do worse. But certainly we need to be able to capitalize on this identification of patients with matched therapies to overcome that mechanism of resistance. The TGF-beta story, if one subscribes to this subset of these two major subsets of CAFs, these TGF-beta-driven versus this IL-1-driven ICAFs, a lot of the data is suggesting that the TGF-beta CAFs are associated with resistance to treatment, but certainly the ICAFs express a variety of chemokines and cytokines that one would expect would also be contributing to resistance to treatment as well. And in this paper from Fig and colleagues in PNAS in 2013, they either depleted FAP-expressing cells from a murine model of pancreatic cancer, or they blocked CXCR4, meant to block CXCL12 signaling emanating from these CAFs, and showed that really paired quite well with immune checkpoint blockade and probably had at least additive and maybe synergistic effects. And so this really suggests that perhaps targeting ICAFs might be beneficial. And so we're seeing some different stories emerge regarding the potential clinical significance of these subsets of CAFs. And in the work from Turley and colleagues, these ICAFs really represented a smaller contribution of the CAFs in the microenvironment, potentially a smaller contribution over time as well as tumors evolve. And so the subset that's going to be more important to target in the clinic, or whether or not we're going to need strategies to target both of these major subtypes of CAFs, is something that still needs to be determined. So many remaining questions. What's the switch that leads to the accumulation of these suppressive CAFs in the tumor microenvironment? We've already heard from Dr. Jaffe that cancer cell intrinsic signals likely in part drive this process. but when this is intervenable remains to be determined. Are CAF subsets with distinct phenotypes and functions derived from different cellular sources, or do these represent different cellular states? Probably a little bit of both. Are MyCAFs and ICAFs more responsible for resistance to anti-tumor immunity? And perhaps most importantly, at what level can CAF-mediated immune checkpoint blockade resistance be targeted in the clinic? So I'm going to stop there, and I'm going to turn things back over to Sean.
Wonderful. Thank you so much, Dr. Galsky and Dr. Jaffe, for those fantastic and engaging presentations. We are now going to take some of the questions that have been submitted by our online viewers. So a reminder to those that are watching us live that you can still submit your questions. Just click the Ask a Question tab to the right of your slide window, type the question into the message box, and then click the Submit button. That will come through to me, and I'll be happy to read your question to the speakers today. So the first question I'm going to come to is actually a very quick one for you, Dr. Jaffe. There were a couple of people who were interested in finding that reference that you've mentioned from Nature Reviews Gastroenterology and Hepatology. Do you know the reference, or at least the year, so that they can look it up?
I could check while we go to the next question.
Okay, no problem. So Dr. Galsky, I'll come to you then. Is there evidence that understanding immunosuppressive mechanisms in the tumor microenvironment enables more successful combination therapies? And are there gaps in our understanding of immunosuppressive mechanisms in the tumor microenvironment?
So I'll take the latter one first. I think there are certainly major gaps in our understanding, and I think one of the gaps, which probably relates to the first question, is which of these mechanisms are redundant versus which aren't. You see individual papers from independent groups, and we're certainly as guilty of this as anyone, highlighting a specific mechanism, and then there are all these potential mechanisms of resistance out there, but how these mechanisms of resistance are related to one another, and if there's a point of intervention that can overcome a master regulator of resistance, I think that's what we need to understand better.
Fantastic. Dr. Jaffe, could I come back to you for this question as well, and I can repeat it for you. The viewer is asking if there's any evidence that understanding immunosuppressive mechanisms in the tumor microenvironment could enable more successful combination therapies, and also are there any gaps in our understanding currently of immunosuppressive mechanisms in the tumor microenvironment?
Yes, I mean, as it was pointed out by both of us, it's quite a complex environment that we're starting to better understand. And, there are a couple of points. One, when you take a biopsy of one piece of the tumor, it may look very different from another piece of tumor. One may have less suppressive cells or a different phenotype of suppressive cells. Our new technologies are enabling us to really type the different suppressive monocytes. We used to think of M1 and M2. Now we know that there's many different subpopulations based on the cytokines they express and the integrins and other chemokine signals that they express. So I think we have a lot more to learn before we're going to have a good answer to that question, but it's a very important one. I also did find the reference. It's from 2019. It's volume 16, page 282 to 295.
Fantastic. Thank you so much for that, Dr. Jaffe. Let me stay with you with another question. Has anyone looked at effects of intestinal dysbiosis on CAF development and or differentiation? Are you aware of any studies?
I am not. I don't know about you, Matt.
No, it's a great question and something that we've of course been thinking about. It's a great question.
Excellent. Okay, so the next question I'm going to come to. This is actually for both of you. Matt, Dr. Galsky, let's start with you. Have there been preclinical or clinical studies evaluating the efficacy of anti-MAPK or anti-STAT3 signaling inhibitors, specifically in subgroups of pancreatic or bladder cancer patients with chemotherapy-resistant stromal signatures? And if not, do you think these approaches are indicated, or rather would others be indicated instead?
So in bladder cancer, not extensive studies yet, although there has been one study with anti-STAT3, but it was not in a molecularly selected population, so related to the aspect of the question, are we selecting the right patients for these approaches? I think that our understanding of these signatures has probably outpaced a little bit what we're able to test in the clinic in a CLIA certified manner with a diagnostic that meets the criteria that would be required for patient selection in the clinic, and this is a common dilemma. And so, yes, some of those combinations have been tested, but perhaps not in the right population, subpopulations of patients.
Right, Dr. Jaffe, any thoughts?
Yeah, so I think these are really important thoughts because STAT3 clearly is important for reprogramming these monocytes. There's been significant data in the literature showing this. STAT3 has been hard in the past to target clinically, but I believe that there are a couple of companies that now have good molecules for this, but it has not been tested in the clinics yet. It is being tested preclinically in pancreatic cancer. And as far as MAPK inhibitors, I'm not aware of anything in the clinics in pancreatic cancer.
Perfect. Next question. Let's start with you, Dr. Jaffe. CAFs seem to influence immune responses to tumors, but is there any evidence that tumor-infiltrating T cells or T cell subsets can influence CAFs or modify CAF phenotypes?
Oh, that's a great question. I'm not aware of T cells specifically influencing them, but I think that one of the problems we have is we can't, particularly in humans, it's very hard to get multiple time points, and I suspect that, I know it's a dynamic process in the tumor microenvironment, and it is quite possible that infiltrating T cells may be secreting cytokines that alter CAFs, eventually leading to T cell exclusion.
Dr. Gelsky, did you have anything to add?
No, I'm not aware of any specific data regarding T effector cells. Certainly there's a suggestion that there's potential crosstalk between Tregs and CAFs, but I do think probably more in the direction that we were talking about before versus the direction of the question, which is Tregs to CAFs. So interesting question that I don't think we know a lot about.
Perfect. Thank you so much. So one last question, I think, maybe we'll squeeze in two. Dr. Gelsky, I'll give this one to you. Did you see any relationship between CAFs and other cells of the immune system, such as tumor-associated macrophages or dendritic cells?
So at the bulk level, you see most of these traveling together, and that's probably not surprising because that's where a lot of these immune cells are located, is in the stroma. And so, identifying these tumors that are really rich in one immune subset versus another, at the bulk expression data, we've never been able to do that quite well because all of these cells really travel together. Now, there are subsets of cells, and there's probably spatial localization of these cells that are important that you don't really get from bulk data, and that's why, of course, single-cell data, in addition to the newer technologies for imaging cellular subsets in their localization and spatial context, that's really going to be, I think, informative in understanding how these cells travel together.
Okay, fantastic. And let me just give you one last question for both of you. I know it's maybe not a short answer, but do your best. Do you have a sense of how much variability there is in immunosuppressive mechanisms present among patients with the same cancer type? And if there is a significant variability, does it make sense and is it feasible to account for this in a therapeutic approach for a particular individual? Dr. Jaffe, maybe I'll start with you on this one.
Yeah, the only data I know is from the TCGA data, and we actually published this and also looked at this in specimens we had. These are from primary pancreatic cancers. And it was very interesting. It's a good question because we saw about 15% of patients had expression of checkpoint inhibitor molecules. However, they varied. So nothing was more than a few percentages. There were some co-expressions, but that was not the predominant phenotype either. And we have published this last year. So you're right. What would we do clinically? So I think our clinical trials are moving toward precision immunotherapy in this realm as well, where the goal would be to have a pretreatment biopsy and to pick immune checkpoint inhibitors based on that. I know that there is one study that I believe is open where there are six or eight arms. Bristol Myers Squibb has a study which I helped design years ago that's now open, and based on the checkpoints expressed, depends on what arm you go on to. So I think that's the beginning of trying to utilize patient-specific information to choose immune checkpoint inhibitors.
Excellent. Dr. Galsky, any last thoughts from you?
Yeah, those efforts are certainly ongoing. There's a trial that I believe is being supported by the Parker Institute that's simply looking at CD8 T cells by immunohistochemistry and determining whether or not starting with that information can help separate patients into different buckets, so to speak, who might benefit from certain treatments versus others. There's an effort from the National Cancer Institute to design a large study that would really have this focus as well, with multiple arms based on immune phenotyping patients' tumors at baseline. And so lots of movements in that direction, and it'll be a little bit of time before we understand if those are successful.
Great. Well, unfortunately, that's all we have time for in this broadcast. We've run a little over time, but thank you to the audience for staying with us. A very big thank you to our speakers today, Dr. Elizabeth Jaffe from Johns Hopkins University and Dr. Matthew Galsky from the Icahn School of Medicine at Mount Sinai. Please go to the URL that is now at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This webinar will be made available to view again as an on-demand presentation in about 48 hours. If you'd like to receive alerts about future webinars, you can go to the sign-up link in the Resources tab just to the right of your slide window. We're interested to know what you thought of this webinar. Please send us an email at the address that's now up in your slide viewer, webinar@AAAS.org. Again, thank you so much to our panel and to Cell Signaling Technology for their kind sponsorship of today's educational seminar. Goodbye.